Okay, so hello everyone. Before I start, I would like to acknowledge the Gadigal people of the Eora Nation, who are the traditional custodians of the land from which I am sitting today, broadcasting the seminar, and pay our respects to their elders past, present, and future. So today I'm delighted to introduce Dr. James Clugston, who's a postdoctoral fellow currently at the Royal Botanic Garden Sydney, working on phylogenomics of a group of legumes. And James has been with us previously also as a PhD student working on cycads. So James today decided to give a talk on a topic that is really important to him, which is about bridging horticulture and science. And so we're really excited to hear from you, James. Off to you.
Okay, well, Hervé's introduced me pretty well, but I'll give you a bit of a background kind of thing to why this is so important for me. I spent the early part of I'll say when I was a young student, based at the Royal Botanic Garden in Edinburgh, where I did my degree and I did my master's degree and part of my PhD as well. And the one thing that Edinburgh taught me was the importance of living plant collections, so in a botanic garden. So of course, a botanic garden, we go there a lot of the times to look at plants. But of course, there's aspects of science, there's aspects of many herbariums and things are based there. So there's a real strong links with history and things. And this is really what I want to bridge on here now. And it's important we have a good connection between the science side of a botanic garden and the horticultural side of the botanic garden. So it means that so we have living collections that have a good emphasis on both science and horticulture here. So most of the images here will be Australian stuff. There's a couple of images here of Edinburgh because I'll be honest with you, since I started this position last year, it's not always been the most opportune time to take pictures in the actual gardens themselves. And if I could get access to the garden at the moment, I would go around and take some pictures to fill in this slide. But because of COVID and things, we're a little bit locked down. But living collections, in my eyes, are probably one of the most essential parts of a botanic garden. This is really what draws visitors. It's great that we've got a good science and things, we've got herbarium, but that's not necessarily what draws the visitors. The visitors go there almost like a zoo to look at these plants as a place of recreation. And botanic gardens really do inspire, and they inspire infrastructure as well. So we can look at the palm house just at the back, just here. This is Edinburgh. This was built in, I think it was 1848. It was older than the Kew Glass House. It's the tallest one in the UK. And we think about this infrastructure, and what was this infrastructure created for? It was to grow plants. This infrastructure was designed and inspired to grow plants that we couldn't necessarily grow outside. And botanic gardens, you see, really pioneered early plant collections. Without botanic gardens and people that were interested in botanic gardens, although the first botanic gardens, if we think about the very old ones, say the Chelsea Physic Garden, which started around, I think that was about 1380. This is as old as a garden goes. And this was originally a medical garden for growing medicinal herbs. And eventually these became more and more into actual botanic gardens as we see them now. The botanic gardens pioneered the collection and the finding of plants through very early plant collectors. And it's probably the best place to see rare and exotic plant species and also to learn about these plant species with really good interpretation. We do this very well in Sydney with the calyx of different exhibitions that happens there. For example, the carnivorous plant exhibit, which was a couple of times ago, was just excellent and very well done. And I think that this is something that we can work on. So really, what are the purposes of botanic gardens? So of course, it's of public interest. They're used for recreation. They're used as a place for education to educate. And they're used for horticultural and scientific research. And botanic gardens are a great way where we can use them to communicate science. And more recently, botanic gardens have started to push in the direction of conservation and put forward conservation stories. And now you can know that in times of bushfires and climate extremes, the conservation of these wild plant populations are becoming more and more important with many species. And this is important that we consider this. So as I was saying, really, botanic gardens are moving in a different direction, are really moving towards the conservation aims. And this is really because the threats that are posed on natural populations by human medicated activities, removal of plants from the wild, eruption of roads, illegal poaching and harvesting from wild populations is becoming increasing more and more. And with this, it means that the conservation concern for many of these plant species is only going up all of the time. And because the conservation concern is going up, it means there's a high risk of extinction of these plants in the wild. And sometimes conserving these plants in the wild populations is not always possible, because sometimes these plants could be extinct and might only be present in botanic gardens. But if we want to look at these plants, we want to conserve plants for the future, it's important to consider this, and it's important to consider and having living collections that have both are visually appealing, have horticultural value, but also have scientific purpose as well. So these plants can, these ex situ populations, when I say ex situ, I mean outside of the wild, in situ being in the wild, that they can hold genetic diversity from wild population or genetic reserves. As a background just here, this is some of the Encephalatus collection at Kirstenbosch National Botanic Garden in South Africa. And cycads are probably one of the main icon groups for plant conservation, because out of all organisms on the planet, more than 62% of cycads are either extinct in the wild, extinct, or are endangered or critically endangered, which is the highest organism on the planet for those extinction risks, or group of organisms. So going on to the botanic garden side of things and that. So one of the questions that we often think about is, can botanic gardens hold genetic diversity of natural populations and things? And this is something what we have to think about. So we have to think about, if botanic gardens are going to conserve species and hold representative taxa for species, this is a good figure just here. So I'll explain this figure just here. So this is the number of individuals, and this is basically indicating gene capture, so the capture of that genetic diversity in the wild population. So this is a friend of mine named Patrick Griffith from the Montgomery Botanical Center in Miami, which are very much focused on palms, cycads, and ex situ conservation. So with just over 200 plants, their collections were able to hold up to about 78% of the genetic diversity. So you can see, the more plants you have and the greater it's captured, the more likely you are to hold that genetic diversity in situ conservation, ex situ conservation, you see, and be able to use that as a potential reserve if we need to replenish wild populations. And I've seen, in many cases, when I've spent some time in Kirstenbosch, I spent some time working with their collections and things as well, and also working on some conservation trips where I'm trying to remember the species, but it was a species of Erica that was extinct in the Western Cape. And it's extinct in the wild entirely, and it was only grown in some small fragmented populations in cultivation only. And this was, they knew the complete genetic diversity. There was individuals from Kew in the UK and from South Africa, and they put them back into the wild and seeing how it's kind of going and things. I'm very hesitant how well that works, but it definitely works. But this shows that botanic gardens can definitely hold genetic diversity for wild populations and can be used for in situ. And one of the things that's good in this, if you've got a really good plant collection and a good collection of plants, it's a really good way to educate the public and students and things on the extinction risk of species through botanic gardens. And it's much more easier to do that in a botanic garden than it is doing that in the field. So what about for Australian species? So I can tell you with a case study with a species that I worked at during my PhD, a species called Cycas calcicola from the Northern Territory of Australia. This was a conservation genomic study. So we looked at whole genome diversity throughout the whole genome of this plant. And we wanted to understand what its genetic diversity like. What we found, it was really low genetic diversity. there was evidence of recent decline in the populations visually. So where we thought there was, we looked at, and we used records, Australian Living Atlas of Australia, to look for where these plants have been collected previously. And what we found is that many of these populations we tried to find were no longer there. There was either a road there, or there was some kind of town, or it's just been burnt down and there was nothing there anymore. And you could walk for ages in these areas or in farmland, and you wouldn't necessarily find these plants. So we could see that from the last conservation assessment that happened of these plants, there's been a significant decline. And what we found is ex situ diversity does not represent the wild diversity. I've got a plot just here. So this plot is what we call a structure plot. So each individual block just here indicates a population. So we have 1, 2, 3 populations in the orange, and 4 or 4 populations in the blue, and these mixed ones just here. So there's a little, each one of these contains a block, and a block is an individual. So what we see here, this is from one area. This is in the Litchfield National Park, and this is in the Catherine Reef. You can see a little blue over here. So the 2 colors indicate 2 different genotypes. So we have roughly 2 genotypes here. So we have a good, what we call a good separation in this species. But you see these ones just here. These ones are from 2 other areas and things, Daly Ribbon, Spirit Hills, and the Northern Territory. And this entire block is what is represented in cultivation. And actually, so we have populations from this Catherine region, and we have Daly Ribbon, Spirit Hills, but we have nothing from the Litchfield National Park. We know that just by collecting, but then when you look at the genetic diversity as well, there is a distinction there. And that's what's important to notice. So with this one species, there's a whole section of genetic diversity that's not in cultivation. So if this species and these populations would get removed within the next 10 years, there's nothing to really work around that. So it's our job as botanists or as horticulturalists to consider this and think about how we can hold this genetic diversity or collect seeds from these wild populations and store them in botanic gardens. Of course, one thing that's done very often is seed banking. And seed banking is a good way to hold diversity of seeds, but it doesn't always work with all plant groups at least. So why do we not always use seed plants? Well, it's not suitable for all plant groups. First of all, some seeds can be what we call recalcitrant. So these seeds don't store for a long period. This is a good example with cycads and also a good example of a lot of palms. So with an example of a cycad seed, the seed itself is only viable for about 6 months to a year. And even if you dry that seed, which generally destroys it, and you try and store that seed, that doesn't change. But of course, where we have grasses and we have things like magnolias and other plant groups, we can store those seeds for potentially hundreds of years in the correct seed banking thing. So we can hold diversity quite easily in seeds. But for some plant groups, mainly older plant groups, we can't do that because the seeds just don't store. So that means is that it's harder for us. So we have to grow these plants to conserve them. But the problem is that seeds are great and seeds can be interesting, but I don't really think seeds really draw the public and not necessarily the best resource for education, where having a living plant there, if you want to learn about plants, having something that's living, you can touch, you can feel, and you can observe, you can smell, that's what makes it really a good draw for the public. Also, having living plants there means we can get access to direct genetic material in the terms of leaves and for taxonomic and systematics research, or collecting cones or collecting flowers and pressing those flowers. And it's a really great way of being able to communicate science very well, is having these plants physically there and creating really good, solid interpretation boards to say, okay, this is what we've got. This is what's research is happening in this collection. This is what these plants are. And this is what we're doing to help conserve these plants. And this is really cool because, etc. And without living plants, we can't really get really good photography and really good video to better, for me especially, to kind of help express our scientific interest or our interest and communicate our science for these plants. So with the Royal Botanic Gardens, when I say the Royal Botanic Gardens, I'm referring to Sydney, Mount Allen and Mount Palmer. There's been an evident disconnect between science and horticulture ever since I've been at the garden. And I'm not saying that that necessarily is always the case of everyone, but it seems to be a little bit the case. And I was used to coming up from gardens like Edinburgh that have a very strong connection between science and horticulture, and often working in the field to do multi-collection trips between science and horticulture, and even people from education going into the field to collect plants, focus on different aspects and things, which was very important. And so at RBG, we really need to build a stronger connection and collaboration between our science side of the branch and horticulture. And this will help display our collections that can be used by science. And one of the things that a lot of botanic gardens are reaching to at the moment is increase the number of plants in living collections of known wild origin. Now, why is this important? If you've got a plant that's been bought from anywhere you see and it's in the collection, it's straight away that plant is good for interpretation. The problem is, you see, is that plant's not always scientifically valuable. If you have a plant that's of known wild origin, you know exactly where it's from in the wild, it means this plant, you can identify it from exactly where it was collected from. And so you know exactly where it was from, what environment it's growing in, and also potentially what population it's from, and so what its genetic diversity holds. So if you're doing research and you're looking at diversity of these plants, by having one that's collected from X and one that's collected from Y, and you find actually, because there's a river that separates these, there's actually a significant genetic disjunction between these, which actually means although there might not be a lot of visual differences between them, they're actually different species because they've speciated on a separate path here. And it's very important to highlight, especially our garden, because from my understanding is Sydney is probably one of the oldest botanic gardens in Australia. I think it is the oldest botanic garden in Australia. And that means we have a lot of historic collections, and it's important to highlight. So these historic collections, although they might not always have the best scientific interest, some of them are very important from a horticultural standpoint. And every plant in a living collection is important for horticulture and has some scientific elements to it, but there's a way we can kind of move forward and increase the potential of these collections. One of the problems we do get in horticulture at the moment, or what there is in horticulture, this is from my conversations with horticulturists like David Lachlan, John Seaman, that I've worked with quite a bit over the last couple of weeks regarding the cycad collections, is we have very few recent additions in the Sydney Garden and Mount Talma that are collected from the wild. And this is something we probably have to work on and change, because if horticulture are going to change their living management strategy to work towards having a collection of more scientific and horticultural value, we have to think that into the future. So how can we improve our living collection? So this is an example where I should have used an image from Sydney, but I don't have one. So this is Dicksonia antarctica growing in Logan Botanic Garden in Scotland, actually. So not really where you would see this tropical looking garden, but it's they get the kind of the Gulf Stream, so they get this like warm stream of kind of water current, so they can grow more tropical plants here. But I would use another image here, but it's still a nice image and it still shows what I want to talk about here. So one of the things I keep talking about is horticultural value. So what do I mean about horticultural value? So there's both on two. So the horticultural value, you see, this is plants that are visually attractive, plants that bring in the visitors, are easy to maintain, are good for communicating, and it draws the eye. It's always good to have collections that you can advertise. Of course, not at the moment because of COVID, but you can bring people into the gardens. And this could be legumes, it could be cycads, it could be any of our hero plants, Banksias, and Willemia. And that's what's we've got to, that's what to consider. So it's always to consider when it comes to thinking about plants, is plants that are valuable for horticulture and are valuable for what the public want to see and what are interested in. But for Australia, there are a lot of species in Australia that are not in cultivation at all and are not represented in botanic gardens. And this is something a bit of a problem, because not everybody can get out there and see these plants, and having these plants in botanic gardens. And even if they are there, people don't necessarily know what these plants are. There are some excellent people out there that go and find plants and use Garden Explorer, use online explorers and use Living Atlas of Australia to highlight these plants and things and identify them in the field. And my hats off to you, they do a great job. But it's still a good way. And Australia has an incredible floristic diversity, and we need to kind of show that off as much as possible. And when we think about what can be used to kind of bring these plants into cultivation and into our collections, well, of course we can use seeds, we can use cuttings. And in science, this is something we have to kind of think about, is what they can use. We often collect plants from the wild. I do this as well. And I don't necessarily always think about cuttings. I do think about seed, but I think about seed as a good scientific instrument. But it's very important to think about, when we're looking for plants, what plants could have this perceived horticultural value and that could be useful for our living collections. And of course, what we perceive as horticultural value and what horticulture do, this is where we need to work better with and have a better relationship with to kind of communicate this. So what about plants for ex situ conservation? One of the things we've got to think about is how research we do in science can help inform conservation. Because if we're carrying out genetic diversity studies, or we're looking at diversity between species, we have a way we can understand this, a way we can influence this. So we understand what the diversity of these plants is like. And that can help inform conservation, whether this is through conservation management plans, where we're looking at diversity, we're looking at the number of individuals in the wild, and then we create a plan to say, this is what it's currently like, this is what's happening, and this is how we need to correct it. And it's really important that in science we consider targeting rare and threatened species, conservation importance for horticultural purposes, to having collections, to build these ex situ conservation collections, and use our own research to be informed by that, and use these plans that are written and created to work this. But this all requires partnerships. And this requires working with different teams and communicating with different teams to watch each team want. And of course, in science, we write our grant applications for groups. For example, myself with Mobilier, the Fabaceae, the Australian peas, we know what we're wanting plant-wise, and we know what genetic diversity is like, and we know the species differentiation. Or we might know the species differentiation, that's still to come. But still, we still have to consider that as much as possible. And it's very useful that in science that we can build these partnerships and figure out what horticulture are looking for, these plants, what can grow in the gardens, and what we can bring from the wild into these horticultural populations and into these collections and grow these plants. If we grow these plants there, horticulture grow them, and then we create some really good interpretation. With peas, you can grow them and you can have flowering plants within a year or so. And to get really good interpretation with that, to show this is what we've done, this is what research we're doing, these are these really cool plants, and this is what we've done to help conserve them. And conservation is not necessarily just having the plants there and in cultivation, it's also about awareness. And so people care about these plants and building a like mentality around them. So one of the things that I really think that we could benefit from doing is collaborating more with horticulture and potentially having joint collection trips. In science, we collect plants usually, I say, because we've kind of not been doing it at the moment, but we do normally about the without the current lockdown in place. But I would probably go into the week, into the field, probably sometimes a couple of times a week, sometimes every other week, to go into there. And I'm constantly looking for volunteers, for people to go out in the field with. And the problem is sometimes, is for me especially, is that it's great because you can go out there with volunteers. People are so willing to help. It's great when you go with a member of staff because that makes things a lot easier. But sometimes you go out there with a volunteer, and I've got some volunteers that work with me that are absolutely excellent. They're excellent at what they're doing. They've got such tenacity for what they're doing, but you feel very bad because you can't actually pay them for that because you don't necessarily have the budget. You can buy them lunch and things, you can get them there. And that's some of the things. But when we're out in the field, we're very focused, especially I'm very focused on finding that species. That's my aim. When I'm out there, I'm looking for something in particular. I can see other plants, but I have a tendency to ignore other plants at time and be very tunnel vision because I want to succeed and find what I want to find. And that's what's always been quite important for me. So having someone from horticulture there that's considering, oh, this is a really cool plant, this could be great for our collections. And we collect a herbarium specimen, collect some DNA, potentially a cutting, potentially some seeds from that plant. And that's very useful to think about. And that's something where we can. Also, it means that we have people to go into the field with as well. So of course, it comes to selection as well. So we're targeting specific plant species, but often in those areas, there are other plant species that have value for both science and horticulture, or have significant horticultural value and could be better represented in our living collections. And I think that really we can work better by working better together with horticulture to help collect these plants from wild populations and bring them into cultivation. Because, of course, we have good examples of things like Wallemia, which is well represented we know the diversity in the wild. We have a good representation in horticulture, but not necessarily all in the wild. Some of you won't necessarily agree with all the points I'm putting across here, but this is always open here. And we've got to think about of these collections. So if we put this work in and work with horticulture, as science, what can we use these collections for? Well, it means we've got a greater diversity of native species. I always remember when I first came to Sydney, I came from Edinburgh. I've spent, and one of my focuses in my career, at least, has been the cycads. And when I came to Sydney, I was so excited to go out there. And I knew Ken Hill that was here previously working on cycads. And I was expecting to see the most amazing Australian cycad collection. But I didn't necessarily always see that, you see. And that was a bit of a disappointment for me when I first came to Sydney. So a lot of the times we don't necessarily have the best displays of natives. We do in some areas of the garden. And of course, this is the exception is the Mount Annan, because that's just Australian collections there. But also having plants, again, of known wild origin. So if you know them of wild origin, it means those plants are incredibly useful for science, and they're incredibly useful for my research and everybody else, because sometimes it's fine to have, say, X species of Pultonea. That's really cool, we've got that. But if we want to verify if it is definitely that species, knowing exactly where it's collected from the wild is very important. And then so of course, in collections, we have something called an accession number. And the accession number is a little tag that links to every plant. And one of the things a lot of living collections have moved towards is having something called a genotypically qualified accession. So it's an accession number. So we say we get a batch of seeds into the collections, and you grow that batch of seeds. When you grow that seeds, if they're from one female plant, those seedlings are all from that same genotype. And in some accession systems, we give them each individual numbers. But when we use a genotypically qualified accession that's used by, say, Edinburgh and Kew and other gardens, we have the same number, but with an A, B, C, or so on qualifier to indicate, so you know straight away that that plant is from that same batch and that same genotype. And really, by doing this, it means we have a greater allelic diversity for many target species in these collections. And if we have these collections as well, and we have really good representation of Australian taxa, it means we can look at flowering, we can look at coning times of these plants and create a really good phenology database. So we can do real phenological analysis and even see if these plants are flowering at different times or coning at different times to the wild, which is quite interesting. And we can collect fresh DNA. That's always important. So what about for education? So really, by building on these collections and working with horticulture, it really will allow us to really build on these collections to help us for the public, for our own interests and for our own research, and also what we want to display at the times as well. And it's a really good way to communicate our research to the public and also to students and even to external stakeholders so we can better engage as well. We have to help create and build on collections that have both horticultural value and have scientific value. So these these collections that aren't just, they can be great looking collections, but they can also have real scientific. A really good example of that is Edinburgh at the moment. Their entire living collections, I think I checked it the other day, and it was at 98.6 or something % of their living collection is of known wild origin. That extra 2% are plants of historical origin. So plants that basically were there before the accessioning policy, big oak trees and things like that. And really, this will allow us to educate the importance of plant conservation and really get the general public interested. So they know that we're not just a botanic garden that looks really pretty, we actually do some really important research in this. And of course, there were ways to work with interpretations and things as well on this, but it's a good way to go. So to kind of conclude this, one of my things that I would say that we could do is we could target research and collection with living collections in mind. So we can always consider what could be good in horticulture and is useful for our own research, that we can just go that little bit extra, collect some seeds and pass them on to horticulture. But of course, we need to kind of communicate that and see what we want between everybody there. I also consider how we can use and contribute to our collections as scientists. And also as from the horticultural side of things, of what if they start collecting themselves, of what they can collect to help with our research as well. And this will allow us to bring us both teams together through strong collaboration, and hopefully to bring new and exciting plants into cultivation. So that's pretty much it, really. I'm happy to open up the board to any questions. If you guys want to email me directly, my email address is here and my website as well, and you can follow me on Twitter as well. I've always got a few questions there. So thank you very much.